Chapter 3, Section 10, Anaerobic Respiration and Metabolic Modularity. Wow, that's a nice name, modularity. Nice word there. Keep in mind, respiration will occur under either oxic with O2 or anoxic, no O2. Okay, it can occur under both. It's just that if you're aerobes like us, it has to have oxygen there or everything shuts down. Those organisms that are anaerobic, oh, there better not be oxygen there. Oxygen's actually poisonous. They're going to use other things, nitrogen-containing compounds, sulfur-containing compounds. What's interesting is the organisms that straddle this. We refer to them as facultative anaerobes. If oxygen's present, cool, I'll use it. Oxygen's not present, cool, I'll use something else. E. coli is a great example of this, because think about it. E. coli, Escherichia coli is an enteric bacteria. It means its normal place to be is in the intestines of mammals. Low oxygen levels there, but you played with it this week in lab. It's on an agar plate. It's exposed to oxygen. Doesn't seem to kill it. If anything, it actually grows just fine in the presence of oxygen. It utilizes the oxygen. If it's there, it uses it. If not, oh well, I can use something else. Many organisms retain the metabolic functions of some of the early cells. Because you have to remember, originally, our atmosphere had no oxygen. It was methane, it was sulfur dioxide, it was many things other than oxygen. Oxygen wasn't even there yet. So, early things, early cells, those protocells, those early cells, before we became what we would think of as prokaryote, before we, living things here on planet Earth, even had a twinkling of this is what eukaryotic can be. Everything was anaerobic. So, chemoorganotroph, chemolithotroph. Organic, inorganic. Organic, but no electron acceptor needed because all you're doing is glycolysis. That's what fermentation is. If you're going to do the electron transport chain of cellular respiration, what's your final electron acceptor? A sulfur-containing compound or a nitrogen-containing compound? Then you're an anaerobic respirer. If it's going to be oxygen, you're aerobic. You see the same pattern here without the fermentation, though, for the chemolithotrophs. Sulfur, nitrogen, anaerobe. Oxygen, aerobe. It's just, where did that original excited electron come from? Who got oxidized? Was it a carbon-containing thing, or was it anything but a carbon? So as I mentioned before, with certain anaerobes, you can get fermentation and anaerobic respiration occurring in the same cell. Maybe not at the same time, but yeah, the potential's there. The genes are there. Respiration's going to require some form of an electron acceptor, some sulfur compound, some nitrogen compound. Fermentation doesn't. Fermentation just needs NAD and a diphosphate. ATP is going to be generated by substrate level phosphorylation. NAD will get reduced to NADH, which will then power some form of respiration in the cell, the electron transport chain, ATP synthase, whatnot. Talked about E. coli. It's a chemoorganotroph. Can be, the facultative anaerobic part about it. If O2 is there, it'll use it. If not, oh well, can do other things. Grows fastest, as I said, under aerobic respiration. It means that it's going through glycolysis, the citric acid cycle, oxidative phosphorylation, very, very quickly. Uses nitrates as an alternate final electron acceptor if oxygen's not there. And many proteins can be inserted into the electron transport chain if alternate carbon sources, alternate final electron sources, acceptors, all of that is occurring. You still get the same basic, complexes, set of proteins, complex together. You swap out, though, the quinones. You swap out, alternative dehydrogenases and things like that, terminal reductases, depending upon growth conditions. High oxygen, low oxygen, no oxygen. What's the carbon source? Things like that. The predominant carbon source. Are we going to be able to go through lots and lots of glycolysis? Or are we going to have to switch over to fermentation and then use that to drive the electron transport chain? Under aerobic respiration, look, you have complexes, and it's not the 4 complexes like we've seen before. It's an abridged set. Cytoplasm, remember the periplasm is the space between the cytoplasmic membrane and the outer membrane. NADs, FADs, just like we've seen from glycolysis and the citric acid cycle. They will be oxidized. Those excited electrons will then be bled of energy past hydrogens pumped. Right there, if it's aerobic, there you go. There's the final electron acceptor. If it's in an anaerobic state, some of the players involved in these complexes will be swapped out. Okay? They're swapped out. And our final electron acceptor is nitrate. Nitrate to nitrite. Oh, kind of cool. Kind of interesting. So another point to point out here. If you stop and think about it, citric acid cycle in eukaryotes occurs in the mitochondria along with oxidative phosphorylation. In prokaryotes, since there's no internal membranes, there's no organelles, citric acid cycle and glycolysis are happening in the cytoplasm in the same space. Hey, look, succinate to fumarate. That's one of the intermediate steps of the citric acid cycle. So instead of having to have the shuttle take them directly from, one part of the mitochondria to another, this is occurring in the same place. So FAD takes it from here right to there. Not saying that succinate immediately interacts with this complex, because it still has to have FAD there. But all of this is occurring in the same place. It's like, as you're watching this, or you're sitting in class as I'm lecturing this, and not only do we have microbiology going on in the same classroom, but we would also have, cell biology being taught in the same room at the same time. It's kind of cool. Then again, I'm a microbiologist. I've been dropped on my head a lot. No complex 3, like you've learned. Okay? Because of this, there's a lot of energy being lost. Second law of thermodynamics. Okay? There's losing a lot of energy there. Not as many hydrogens get pumped for every 2 electrons. So there is, it's not as efficient as what you would see in our mitochondria. And as I said, if no oxygen's present, swap out some parts, E. coli switches over to nitrate. Nitrate reductase. Now becomes part of complex 4, the final reductase. It's a less electropositive than oxygen, so less energy is coming about in play here, but it still works. Oxygen's present, E. coli is going to have maximal ATP made, maximal energy, highest metabolism. No oxygen, switch over, still pretty good, just not as good. Aerobic always conserves more energy. Will always outcompete anaerobic. It just works better.